So in the previous video lecture, we performed differential abundance testing to detect ASVs that were significantly different between dogs G and B. So we started looking at the CSV output, and here as rows we get all our ASVs that were significant between the two dogs. And at the bottom we'll find our model where dog G was coded as 1 and dog B as 0. And then we also have a scaling factor here for each one of our samples, which is used by metagenome-seq to account for differences in sampling depth between our samples. So the top here we have some helpful columns so that we can familiarize ourselves with the data, what the data looks like for each ASV. So percent positive in groups 0 and 1, so that would be dogs B and G. The number of samples that were actually positive in those groups. So, and also then the mean number of counts for each one of those groups. So if we look at this top ASV, and that was for a Sporobacter, and we can go look at what that heat map entry looks like. So here we've got the Sporobacter row, and we can see that 4 out of the 5 dogs samples for dog B had this ASV, whereas none of the dog G samples had this ASV. So if we look again at the stats, we'll see that it did have a significant Fisher's exact p-value that was 0.047, but that was not significant after multiple testing correction. However, the columns we see here after the Fisher's exact test, so the coefficient p-values and adjusted p-values, that is from our metagenome-seq GLM with a zero inflated Gaussian distribution. And we see by that it was significant even after adjusting for multiple testing. And the coefficient here gives us the size of that difference between the two groups. We can have a look at another example. If you look here at an ASV like Prevotella copri that was present in all our samples, but definitely more abundant in dog G. Let's have a look at the stats for that. So here we see Prevotella copri is down here. We can see it also had a highly significant p-value and a fold change or coefficient of 3.89. Then here we have sorted by dog, and it's also worth having a look at your other factors of interest. So we see that there's no clustering by treatment. In this case, we haven't really clustered the samples, but if you see any patterns by treatment, then you might want to include that factor as a covariate in your analysis to make sure that the results you're getting is from your differences in dog and not your differences in treatment. But in this case, we can see there is absolutely no pattern here by treatment. I just want to show you guys the effect of this percentage presence parameter. So in our case, we pre-filtered the data to actually take away all those low-level OTUs. So in that case, it's not that important. But say we hadn't done this pre-filtering. So I've gone and done that as an example. I just take the raw data, give it to this function, and leave the parameters as is. Then we actually get 59 ASVs that are listed as significant instead of the previous 40. And if we look at that heat map, we'll see that there are a whole bunch of ASVs at the bottom here that was only present in one sample. And I really wouldn't be comfortable in calling that significantly different between dogs G and B just because they were present in this one sample. So we are working with really small sample sizes here. We discussed that there are other methods like EdgeR and DSEG2 that might perform better at such small sample sizes. But what we can do here is make sure that our filtering is good so that we only end up with quality results. So if we hadn't pre-filtered, then we could have maybe increased this percentage to 0.6 or 60%, which requires at least 3 of the 5 samples in either one of the groups to be positive. So if we rerun, we can see what that does to the results. We can see now we're back to 40 ASVs. And So now it's taken out those ASVs that were really only present in that one sample. So it is important to pay attention to your results, and that's why it's really useful to visualize it as a heat map. So you can just, visually your eye will pick up patterns and tell you, okay, my filtering is good enough, or maybe I don't really trust this and I have to increase my percent presence to something a little bit more. Lastly, I'd just like to make you aware of an alternative model in MetaGenomeSeq called fit feature model. So the custom function that we use was built around the fit_sig function, and that was quite a while ago, and the authors have since released the fit feature model function, and according to them that is currently the preferred method. So you can go and consult the MetaGenomeSeq help file on bioconductor.org, and I think that would be worth looking into, and future versions of our microbiome custom functions might include this. So I've listed here on the H3A BioNet downstream analysis page. You'll see at the bottom here we've got the microbiome custom functions R-gist, and that is where if I do any updates to these functions, you'll be able to find it here. And then one last thing is that when we did our normalization, we used what we call standardized counts, and they were calculated based on relative abundance, and then we multiplied those relative abundance by the median sampling depth. And we use that for our beta diversity analysis and our bar plots. But one alternative would be to use the mrcounts function from MetaGenomeSeq, and that is what they use for the normalization. So those scaling factors that were included in the model, you can also apply that to your raw counts. So you apply it to an MR experiment object. So we would have gotten that using the make MetaGenomeSeq function that we applied to our phyloseq object, and then you would say normalize is equal to true, and then that output will give you normalized counts that you can use in other aspects of your analysis. That brings us to the end of this module for 16S downstream analysis. I really hope that you all enjoyed the lectures and that you now have a good foundation on which to build for your own analyses in future.